So good day, dear doctors, and welcome to this very important and high yield coaching session, which will cover the essentials of clinical biochemistry, and we will summarize all the must-knows regarding amino acids. So I'm Dr. Toom. I am your guru guide, and let's crack the board exam and your biochemistry exams together. Now, I want you guys to take reference that what I'll be using for this module is going to be one of the most high yield references for biochemistry. And this is, of course, Harper's Illustrated Biochemistry. Now, no matter what edition you're using, biochemistry concepts has not changed. So this is basically chapter 3 of amino acids and peptides. Now, we're going to start with the biomedical importance of amino acids. Now, amino acids are very important because amino acids are the building blocks of proteins, okay? Amino acids provide monomer units, which will basically comprise polypeptide chains of proteins. Amino acids also participate, okay, in several cellular functions, particularly, number one, nerve transmission or neuronal transmission, the biosynthesis of important biochemical products such as porphyrins, which of course is very, very essential and very important for heme synthesis. Very important is also the role of amino acids in the synthesis of our purines and pyrimidines, as well as urea. And amino acids also play a very important role as peptides, which are part of hormones. So they play a role in the neuroendocrine system. Now we're going to introduce this term, which everyone must bring with them to any biochem exam. And this is the term essential amino acids. Now, what do we mean by essential amino acids? Okay, so again, what do we mean by essential amino acids? So when we say essential amino acids, these are amino acids which cannot be synthesized by the body. They cannot be synthesized by the human body. Therefore, they have to be provided through the diet. Okay, now the textbook tells us there is an estimated 10, okay, amino acids which are essential. Okay, so please take note of this. Now, let's dissect the textbook. The textbook mentions some of the biomedical importance of amino acids. And one of them is the property of microorganisms or bacteria to secrete amino acids or peptides. And one of the examples mentioned in the textbook is bacitracin. Now, bacitracin is an antibiotic. It is a very common antibiotic which is used in our antibiotic sensitivity. Okay, the mechanism of action of bacitracin is to inhibit cell wall synthesis. So if it inhibits the cell wall, particularly of gram-positive organisms, this is how it exerts its effect, and this is how the bacteria with a gram-positive cell wall will die. Okay, now another antibiotic mentioned in Harper's is gramicidin A. So what was the first? It was bacitracin. The second is gramicidin A. Now the mechanism of action of gramicidin A is to interrupt protein function and to interrupt DNA function. So again, bacitracin inhibits cell wall synthesis. Gramicidin interrupts both DNA and protein function. Now, there is an anti-tumor or an anti-neoplastic drug which is mentioned in the textbook, and it is bleomycin. Okay, now, question. What is the dreaded respiratory side effect of bleomycin? What is the dreaded side effect? Okay, the dreaded side effect of bleomycin. Okay, very good. Bleomycin is notorious for causing pulmonary fibrosis. Now, can anyone tell me what other drugs can cause pulmonary fibrosis? So again, what other drugs can cause pulmonary fibrosis? So there is another anti-neoplastic or anti-tumor drug, which is busulfan. And we have an anti-arrhythmic, which is amiodarone. Now, for the biochem exam, okay, I'm specifying this because Harper's only mentions bleomycin. When you read your pharma books and your medicine books, it adds busulfan and amiodarone. So please take note of these three very important drugs, okay? Now, there's a mention also in Harper's of microcystine and nodularine. Again, microcystine and nodularine. Now, what are these two substances? These are considered to be cyanobacterial peptides. So these cyanobacterial peptides, when they are present in large doses, they can cause liver tumors. They can cause hepatic or liver tumors. Now, another very important amino acid, okay, are those that are found in the seeds of legumes, particularly the genus Lathyrus. I'll explain this in a while because there is a neuroparalytic disease mentioned and in italics in Harper's, which is called Lathyrism or neurolathyrism. Okay, now here is what I want you to remember and what you have to bring with you to your respective exams. Lathyrism is defined as a neurodegenerative disease, okay, which is associated with the chickpea or what we call the grass pea. Okay, the chickpea or the grass pea. So please take note of this. Now, the classic presentation of neurolathyrism is one of paraplegia, which means there's paralysis of both lower extremities, there's tremors, and there's muscular weakness. Now, what important neurodegenerative disease classically presents with tremors, particularly resting tremors? What important neurodegenerative disease presents with resting tremors? Okay, so it is Parkinson's disease or what we call PD. Okay, Parkinson's disease or PD. So grass pea or chickpea. So here is the seeds, okay, of the legume, and here is the flower. Now, this is highlighted in your textbook, the Cycad seeds. Okay, these are the Cycad seeds, okay, which you see here. This is a Cycad, and it has seeds which contains neurotoxins. Now, these neurotoxins, okay, are very important because they are risk factors. Okay? They are risk factors for neurodegenerative diseases. And there are two neurodegenerative diseases mentioned in the textbook. Number one is ALS, or amyotrophic lateral sclerosis, and number two is PD, or Parkinson's disease. Memorize this for your exam. So what are the two neurodegenerative disorders which arise from ingestion of Cycad seeds? It is amyotrophic lateral sclerosis, or ALS. Then we have Parkinson's disease. Now, please take note. In the textbook, they mention a complex, which is the Parkinsonism dementia complex. This is because of the neurotoxin, okay, L-beta-methylaminoaline. Okay, so please take note of this, B-M-A-A. So it's the L-beta-methylaminoalanine. Okay, B-methyl aminoalanine. This is the specific neurotoxic amino acid, which is found in what food or what fruits? It's found in the cycad seeds. Now, it is also discussed in our mother textbook that it is found in a specific population, okay, the natives of Guam, who have a habit or a practice or culture or custom of ingesting either the cycad seeds or fruit bats that feed on the cycad seeds. So for your exam, memorize BMAA, memorize cycads, memorize ALS, and memorize your Parkinsonism dementia complex. Now, this is a table lifted from our textbook. They're going to mention the toxic, okay, alpha amino acids. We have homoarginine. We have ODAP. Okay, just memorize ODAP. Okay, then we have BAPN. Then we have the diaminobutyric acid. And lastly, we have here the BMAA. So here is the BMAA. So it says here it is a possible risk factor for neurodegenerative diseases, particularly ALS and Parkinsonism dementia complex. Now, the diaminobutyric acid inhibits ornithine. And always remember, ornithine is found in what biochemical pathway? Ornithine is found in what biochemical pathway? It is found in the urea cycle, okay? So problems with ornithine or inhibition of ornithine transcarbamylase, which is one of the important enzymes for the urea cycle, will present with ammonia toxicity. Now, the BAPN is an osteolarythrogen, meaning it affects the bones. So we have here the prefix osteo. ODAP is associated with neurolathyrism, and homoarginine is also associated with neurolathyrism. So one more time, what are the 5 potentially toxic L-alpha amino acids? Homoarginine, ODAP, BAPN, BMAA, and diaminobutyric acid. So here is a photo of the legumes. Okay, so this is one of the potentially neurotoxic amino acids, okay, which causes neurolathyrism. Here's the ODAP, okay, and here's the homoarginine, which we mentioned in the previous table. And we have here the BAPN, which is actually an osteolarythrogen. This is from the sweet pea, the flower of the sweet pea. So please remember, you are not attending a lecture. You are attending a coaching, which means we have the presumption you have already touched this from the mother textbook. Now, regarding the alpha gamma diaminobutyric acid, it is an ornithine analog. So it inhibits ornithine transcarbamylase, which is an important enzyme in the urea cycle, and it's going to present with the clinical feature of ammonia toxicity. So here, the cycad seeds, remember, amyotrophic lateral sclerosis and Parkinson's or Parkinsonism dementia complex. Okay, now there are some naturally occurring amino acids which we have to bring from this chapter. Number 1 is the one we find in the brain tissue. So what are the naturally occurring amino acids that we find in the brain tissue? So we have serine, then we have aspartate. Okay, again, we have serine and we have aspartate. We also have some naturally occurring amino acids which are found in gram-positive bacteria. One is D-alanine, the other is D-glutamate. So D-alanine and D-glutamate. Later, when you take up microbiology, you will realize there are some antibiotics, for example, vancomycin, which would basically affect these amino acids. There are also some gram-positive bacteria that has a capsule. Okay, it has a polyglutamate capsule. Now, curious, what is that famous bacteria? What is that famous bacteria that has a poly-D-glutamate capsule? In fact, it is the only bacteria that has a poly-D-glutamate capsule, which increases its virulence and increases its pathogenicity. Okay, very good. It is Bacillus anthracis. And what disease is caused by Bacillus anthracis? So what famous disease is caused by Bacillus anthracis? It is anthrax. And anthrax comes in three forms. The most common, you have the pulmonary, then you have the cutaneous, then you also have the gastrointestinal. Okay, so this is just a clinical application of anthrax. Okay, next, there's another bacteria mentioned in the textbook. This is Bacillus subtilis. Now, this bacteria, Bacillus subtilis, excretes the following amino acids. We have methionine, we have tyrosine, we have leucine, then we have tryptophan. Okay, we have MTLT, Bacillus subtilis, okay, excretes the following amino acids. So what is the significance of the secretion of these amino acids? Okay, this will trigger the biofilm disassembly. Okay, biofilm formation is an important property of several bacteria, which increases its pathogenicity. So this is Bacillus subtilis. Now, there's one more bacteria mentioned in the chapter in the book, and it's called Vibrio cholerae, which of course is the culprit that causes the famous, okay, infectious form of diarrhea, which presents with voluminous, okay, watery stools, and this is Vibrio cholerae. So what Vibrio cholerae does is it incorporates the following amino acids, leucine and methionine. It incorporates these two amino acids into the peptide component of the peptidoglycan layer. Now you have to remember, gram-positive bacteria have a very thick peptidoglycan layer, and this is responsible for giving the gram-positive stain of that bacteria. Gram-negative bacteria, on the other hand, such as Vibrio cholerae, have a thin peptidoglycan layer. Okay, so please remember that. Now, question, what amino acid is the precursor of thyroid hormone? What amino acid is the precursor of the thyroid hormone? And this amino acid, when metabolized, will produce epinephrine, norepinephrine, and dopa. Okay, so the amino acid is tyrosine. Now, question, from what amino acid, okay, from what amino acid does tyrosine come from? What is the precursor? Okay, what is the precursor of tyrosine? Very good. It is phenylalanine. And what is the enzyme that catalyzes this famous reaction, which converts phenylalanine to tyrosine? What is that rate-limiting enzyme? Okay, it is phenylalanine hydroxylase. Okay, phenylalanine hydroxylase. So please remember that by heart. So if you have an inborn error of your amino acids, one of the diseases that can occur is PKU or phenylketonuria. Okay, phenylketonuria. Now, question, what is the neurotransmitter which is a precursor of gamma aminobutyric acid, or GABA? What is the neurotransmitter which is a precursor of gamma aminobutyric acid? Okay, very good. It is glutamate. Now, question, what is glutamate? Is it an excitatory or an inhibitory neurotransmitter? What is glutamate? Okay, very good. It is an excitatory neurotransmitter. So glutamate is the precursor of GABA. Now I'm going to write this equation here. Glutamate is catalyzed to become GABA. Now question, what is GABA? Is GABA excitatory or is it inhibitory? What is GABA? Makagaba. Okay. So GABA is inhibitory. Very good. Now, question. What is this famous enzyme that catalyzes this reaction? What is the enzyme that catalyzes, okay, the decarboxylation of glutamate to become GABA? Okay, this is the famous GAD. Okay, so glutamate decarboxylase. Always remember that for a standard biochem exam. Now, let's go back to the basics and must-knows you have to bring with you to any exam which involves the concept of amino acids. So we're going to go back to the basic general structure of amino acids. Now, always remember all amino acids have a side chain. The side chain is what some textbooks call the polar side chain. But I just want you to remember the R group or the side chain because we have polar and non-polar amino acids just to avoid confusion. Just use the term R chain or the side chain. Now, every amino acid has an amino group and it has a carboxyl group. I want you to remember, let's start with the easy one. Letter C is carboxyl. This is the COOH. Now, when you are preparing for a biochemistry exam, clinical biochemistry exam, particularly for medicine or for physicians, you don't have to memorize the freaking amino acid structure like you were back in college or in pre-med, okay? This is clinical biochemistry we're talking about. So the carboxyl group is letter C. It's the COOH. Now the amino group is the NH2. Okay, so again, for the general structure of an amino acid, what are the three parts of an amino acid? Number one is the side chain. Number two is the amino group. Number three is the carboxyl group. Now, question. What will determine the classification of an amino acid? Is it the amino group? Is it the carboxyl group? Or is it the side chain? What will determine the classification of an amino acid? Is it the amino group? Is it the carboxyl group? Or is it the side chain? Okay, very good. It is the side chain which determines the classification of an amino acid. Now, we're going to introduce this term called the chiral carbon. Okay, I want everyone to remember the chiral carbon is letter C. It is at the center. It is found at the center of the amino acid structure. So a chiral carbon is the central carbon which is attached to how many substituents or how many groups. Basta chiral, how many groups or substituents is attached to a chiral carbon? It is 4. So the magic number is 4. So always remember the chiral carbon, like what you see here with the asterisk. This is the chiral carbon. This is the center carbon that is attached to 4 different, take note, 4 different or unlike substituents. Okay, so always remember the chiral carbon. Now question, what is the amino acid that does not have a chiral carbon? What is the amino acid that does not have a chiral carbon? Why is this important? Because all amino acids have a chiral carbon except, okay, for glycine, which is considered to be the simplest amino acid. So please remember that. Now here, so the chiral carbon, this is the carbon in which all 4 groups attached are different. It is the chiral carbon that confers optical activity. Therefore, what is the amino acid that does not have optical activity? What is the amino acid that does not have optical activity? Okay, it is glycine. So glycine does not have optical activity. Now, I want you to bring this term with you to your respective exams, the stereoisomers. This is what we call the enantiomers. So these are the mirror images, either stereoisomers or the enantiomers. This refers to the mirror images. Now, Guru Guide from the med guru himself, Dr. All amino acid side chains have one or more carbon atoms, except for glycine. Okay, now we're going to mention the terms dextrorotatory and levorotatory. This is used to describe the configuration of amino acids. Now, all amino acids basically share the same absolute configuration of the mother compound L-glyceraldehyde. So here's the L-glyceraldehyde. When the amino group is found on the left side, like what you see here, this is levorotatory. When it is found in the right side, then it is referred to as dextrorotatory. Now, what is the most common form of amino acids? Is it L or is it D in humans? So in humans, it is predominantly L. Okay, it is predominantly levorotatory. Now, please take note of this. Another guru guide. All amino acids except glycine are optically active. So when you say optically active in biochemistry, it means that that substance or molecule has the ability to rotate the plane polarized light. Okay, so it is glycine which is not optically active. So please remember this. Now, there are some amino acids which plays a very important role, okay, in what we call post-translational modification of amino acids. So again, what is this biochemical process? It's called post-translational modification. There are some amino acids which undergo post-translational modification. So let's summarize what has to be brought to a standard exam. Now, in this chapter in the textbook regarding post-translational modifications, there are two important things I want you to remember. One is collagen synthesis. Second is the blood coagulation cascade. So collagen synthesis. Second is the blood coagulation cascade. So let's start with the collagen synthesis, one of the most abundant proteins in the body. Two very important amino acids are involved in collagen synthesis. This is proline and lysine. Now, these two residues, these two amino acid residues, proline and lysine, has to undergo hydroxylation, and it has to undergo post-translational modifications. So from pro-collagen, it has to be converted to the active form, which is collagen. And this now becomes hydroxyproline and hydroxylysine. Now, question, what important cofactor or vitamin is required for the hydroxylation of proline and lysine? What vitamin or cofactor is required for the hydroxylation of proline and lysine? It is vitamin C or ascorbic acid. Okay, vitamin C or ascorbic acid. Now, the next classic example of post-translational modification involves the blood coagulation cascade. Now, there's a very important biochemical process that occurs, and this is known as the carboxylation of the glutamyl residues. So the glutamyl residues from glutamate, okay? The carboxylation of glutamyl residues will yield the gamma carboxyglutamyl residues. This gamma carboxyglutamate or gamma carboxyglutamyl residues plays a very important role in the blood coagulation cascade. Now, what are the vitamin K dependent clotting factors? So the vitamin K dependent clotting factors, we have the famous 1972. We have factor 2, factor 7, factor 9. Then we have factor 10. These are the vitamin K dependent clotting factors. Now, question: what drug inhibits the gamma carboxylation of the vitamin K dependent clotting factors? What drug inhibits the gamma carboxylation of the vitamin K dependent clotting factors? What is the name of that famous drug? Okay, it is warfarin. Warfarin, or what we commonly call comedine. Okay, now question: what does warfarin inhibit? Is it the extrinsic pathway or is it the intrinsic pathway? What does the drug warfarin inhibit? It inhibits the extrinsic pathway. So remember this mnemonics: WEPT. W-E-P-T. Warfarin is extrinsic, and what you monitor laboratory-wise is the protein. So warfarin, W. E is for extrinsic pathway. PT stands for protein. That is what you monitor if you want to monitor the activity of the extrinsic pathway. So just remember, who made you wept or who made you cry? It is your ex-boyfriend or your ex-girlfriend, basta your ex. And that is why you are at war. Okay, you are at war with your ex. Okay, now, so here, this is the ascorbic acid, very important cofactor, okay, in the hydroxylation of the amino acids proline and lysine. Okay, so if there is vitamin C deficiency, what do we call this condition? If there is vitamin C deficiency, what do we call this condition? This is known as scurvy. And vitamin C deficiency, scurvy, also is associated with poor collagen synthesis. Okay. Now, another guru guide. Make sure you know this. Collagen is a triple helix structure. It has this famous formula, glycine XY, where X is proline and Y is hydroxyproline. So please remember this. Triple helix, collagen, glycine XY, so it's actually glycine proline dash hydroxyproline. Now here, this is the glutamyl residues we mentioned earlier, and this is your gamma carboxyglutamyl. Very important for the blood coagulation cascade, and this is where vitamin K comes in. The drug that inhibits vitamin K is warfarin. Now, another thing I want you to remember. We've mentioned earlier that amino acids are building blocks of proteins. So here's the tyrosine being converted to L-dopa. Then it becomes dopamine. Dopamine becomes norepinephrine. Norepinephrine becomes epinephrine. Therefore, Tyrosine is the precursor of dopamine. It's the precursor of norepinephrine, and it's the precursor of epinephrine. Now, what do we call the collective term for these proteins? Dopamine, norepinephrine, and epinephrine. What is the collective term for these proteins? What is the collective term? Okay, so we call them the catecholamines. Okay, we call them the catecholamines. Okay, now we mentioned this earlier that glutamate is a neurotransmitter and it is a precursor of GABA. So here is the famous glutamate decarboxylase, converting glutamate to GABA. Okay, now let's mention this concept of Zwitterions. Molecules that contain an equal number of positive and negative charged groups and bear no net charge are called Zwitterions. So this is one of the structural properties of amino acids. So amino acids, okay, bear no net charge and have an equal number of positive and negative charged groups. This means amino acids at a physiologic pH of 7.0 exists in a Zwitter ion state. Do not take any biochem exam if you do not know what a Zwitter ion is, okay? Now, so please take note of this. A Zwitter ion is a molecule that contains a positive and negative charge. So it's a dipolar ion, but it is equal. It is equal. No net charge. So please remember that. So that's a physiologic pH of 7.0. If it has an anion state, then that means there's a high pH. Cation state would mean a low pH, okay? Now, another property of amino acids is it is amphoteric. So at a biologic pH of 7.0, amino acids, aside from being in a Zwitter ion state, amino acids are amphoteric, which means it contains both an acidic, okay? It contains an acidic group and a basic group. So the acidic group, this is the carboxyl. The basic group, this is the amino group. Diba? All amino acids have a carboxyl group, which is the acidic group, and it contains an amino group, which is the basic group. So please remember this, amphoteric. So amino acids exist in a Zwitter ion state. Amino acids are amphoteric. Now, this is also mentioned in the textbook, okay? What is used to express the strength of weak acids? This is the famous pKa, okay? Not the pH, not the Henderson's-Hasselbalch's equation, but the pKa. This is the value which expresses the strength of weak acids. So always remember this. Smaller pKa's are the stronger acids. Larger pKa's are the weaker acids. Okay, stronger acids have a smaller pKa, while weaker acids have a larger pKa. Now, this table is lifted from your textbook. Now, tip, you don't have to freaking memorize the numbers. Okay? You don't have to memorize the numbers. So this is just showing you the pK values for the ionizable groups or the dissociating groups in proteins. Example, carboxyl, imidazole, sulfhydryl, hydroxyl, then the guanidinium of arginine. Okay, so always take note, what is the interpretation if the pK is high? Is this a strong or a weak acid? High pKa. Okay, very good. This is a weak acid. Therefore, in this table in Harper's, which is the weak acid, the weakest, which is the weak acid. Okay, which is the weak acid. So it's the one with the high pKa. Okay, it's the high pKa. So in this table, it would be these guys here, these last two, particularly the guanidinium. Okay, now almost done. So another property of amino acids is the solubility. Okay, so always remember the solubility of amino acids will reflect their ionic character. Now, of the amino acids mentioned in your textbook, which has basically the highest, okay, the highest wavelength, okay, the high wavelength to absorb ultraviolet light. Okay, look at the figure. Okay, it is tryptophan. Tryptophan can absorb from 250 to 290 nanometers on spectrophotometry. Now, I want you to remember tryptophan, tyrosine, and phenylalanine. What is common with these three amino acids, which is responsible for their ability to absorb ultraviolet light? What group do they have, or what structure does tryptophan, tyrosine, and phenylalanine have? Very good. They have aromatic rings. Okay, the three amino acids with aromatic rings is tryptophan, tyrosine, and phenylalanine. So here, as you can see, tryptophan absorbs to 10 times more efficiently, okay, ultraviolet light compared to tyrosine and phenylalanine. Now, the R group, okay, remember the R group, okay, the side chain can be characterized either as being hydrophilic or hydrophobic. So it can either be hydrophilic or hydrophobic, meaning the side chain can either be polar or it can be nonpolar. So here again, here's the side chain or the R group. It can either be polar or nonpolar. Now here, I want to highlight the carboxylic acid, the COOH, okay? What is the function of the carboxylic acid groups? They are responsible for the formation of esters, amides, and acid anhydrides, okay? So the carboxyl group, take note of this. So here's where the memorization comes in. So we have the esters, we have the amides, okay? Then we have the acid anhydrides. So please take note of the function of the carboxylic groups. Now the amino groups have the following functions: acylation, okay, esterification, okay, that's for the amino groups. Now for the sulfhydryl and the hydroxyl groups, the main function of these groups is oxidation and esterification. So hydroxyl and sulfhydryl groups is oxidation and esterification. Now, this is always asked in exams. What is the smallest amino acid? It is glycine. So since it is the smallest amino acid, it can be accommodated in places which are inaccessible to other amino acids, okay? And this is usually found in the areas where the peptides will bend sharply. Now, you have to remember proteins have a primary structure. They have a secondary structure. They have a tertiary structure, and they have a quaternary structure. Now, question, what is the primary structure of a protein? What is the primary structure of a protein? So primary is letter P. It is the peptide bond. Okay, the primary structure of a protein is a peptide bond. Now, question, what is the secondary structure of a protein? What is the secondary structure of a protein? The secondary structure is either the beta-pleated sheets, okay, the beta-pleated sheets or the alpha helix. Okay, the alpha helix, the beta-pleated sheets or the alpha helix. Now, sorry to be green, but sometimes we memorize things with green mnemonics. So secondary is number 2. There are 2 people under the beta-pleated bed sheets performing an alpha helix. Okay, now tertiary. This would include the domains, which are very important for your immunoglobulins. It also has the salt bridges. And what is the classic example of the quaternary structure of proteins? What is the classic example of the quaternary or quaternary structure of proteins? What is the example? Okay, the example is hemoglobin. Okay, hemoglobin. Now, I would like to remind everyone, hemoglobin is quaternary, myoglobin is tertiary. Okay, so hemoglobin is quaternary, myoglobin is tertiary, but both are very important complex proteins. Okay, now, the amino acid sequence will determine the primary structure. Okay, so always remember, amino acids are linked by peptide bonds. Now, question, what tests will you perform in the laboratory if you want to test for the presence of peptides? What is that laboratory test which yields a purple or a violet color, which is indicative of the positive presence of peptides? Okay, what is that famous laboratory test? Okay, very good. It's the Biuret test. Okay, a positive Biuret test is purple or a violet color, and this indicates that there is the presence of peptides. Okay, so take note of this statement, lifted verbatim from the book. The number and the order, the number and the order of amino acid residues in a polypeptide. So the number and the order, which is the sequence actually, of the amino acid residues in a polypeptide constitutes its primary structure. Okay, now I want you to bring this to your exams. This is the famous glutathione, which is an antioxidant. Glutathione is very important for the pentose phosphate pathway. Glutathione, okay, intracellular glutathione will protect the red blood cell from oxidative stress and free radicals, as well as reactive oxygen species. Pentose phosphate pathway. What is the rate-limiting enzyme? What is the rate-limiting enzyme of the pentose phosphate pathway, also known as the hexose monophosphate shunt? What is the rate-limiting enzyme? It is glucose 6-phosphate dehydrogenase. Now, always remember the pentose phosphate pathway will yield NADH, okay, or NADPH. Deficiency in NADPH would lead to deficiency in glutathione. Decreased intracellular glutathione would shorten the lifespan of the red blood cell and would predispose to hemolytic anemia. So what will happen when there is a deficiency of glucose 6-phosphate dehydrogenase? The clinical manifestation is hemolytic anemia. Okay, so please remember that. Now, the amino acid sequence of glutathione is gamma-glutamyl, cystenyl glycine. Therefore, if I were to ask you in an exam, what are the 3 amino acids which comprises the structure of glutathione. What would these three amino acids be? Okay, the three amino acids of the structure of glutathione. What are these three amino acids? One is glycine. What are the others? Okay, so we have glycine. What else? What other amino acids comprises glutathione? Okay, so we have glutamate. Very good. We have glutamate, we have cysteine, and we have glycine. Now, these are the amino acids with an aliphatic side chain. Okay, the amino acids with aliphatic side chains. My personal mnemonics here is GAVIL, glycine, alanine, valine, isoleucine, and leucine. Now, the simplest amino acid is glycine. Okay, this is involved in heme synthesis. Okay, very important in heme synthesis or porphyrin synthesis. Alanine is there, valine, isoleucine, and leucine. Now, question, what is the specific classification of these three amino acids? Leucine, isoleucine, and valine. How do we classify them? I call this LIV. So this is the branch chain amino acids. There are three branch chain amino acids. That's leucine, isoleucine, and valine. Now, why am I using the mnemonics LIV? Now, just remember this story by your guru. We branch out to others so that they may live. We branch out to others so that they may live. Now, question, a congenital deficiency of branch chain amino acids leads to what important disease? What important disease? Okay, it is maple syrup disease. Now, in maple syrup disease, these are children who present with hypotonia, mental retardation, intrauterine growth retardation, and they have a characteristic smell of the urine. It smells like maple syrup, or it has a caramel odor. Okay, so another mnemonics here is maple syrup disease, branch chain amino acids. It is like cutting the branches of a maple tree, like cutting the branches of the maple tree. Now, tip, we want to top the exam, we want to excel. Valine is important in what type of missense mutation? Okay, which results to an abnormal shape of the red blood cell. Instead of being the biconcave disc with a diameter of 7.8 micrometers, the RBC will now appear to be sickle-shaped. So this is sickle cell anemia. So what actually happens in sickle cell anemia? There is a missense or a point mutation, which specifically occurs at position 6 of the beta globin chain, where there is now a substitution of what two amino acids? What is the substitution here? The substitution of what two amino acids? Valine is one. What is the other amino acid? Okay, it is glutamic acid. Okay, now, what about the amino acids with a hydroxyl group? OH means hydroxyl. So we have tyrosine, we have threonine, and we have serine. Now tip, tyrosine is not tryptophan. Okay, does tyrosine have an aromatic ring? Yes or no? Does tyrosine have an aromatic ring? The answer is yes, as you can see here. So tyrosine, therefore, has more than one classification. It has an aromatic ring, therefore it can absorb ultraviolet light. It also has an hydroxyl group. Here is the hydroxyl group. Okay, here is the hydroxyl group. Here is the OH or the hydroxyl group. Okay. Now, what about the amino acids that contain sulfur atoms? Now, always remember when it's sulfur, you usually have a sulfhydryl group there, such as this SH here. So we have cysteine, then we have methionine. Now, always bring with you to the boards that methionine is the first amino acid in the genetic code. Now, what do we call the codon, which is the beginning of the genetic code? Okay, we call it the start codon. And the start codon is also known as what codon? What three letters? A, U, G. Okay, very good. I am impressed. And the amino acid which you've identified in the presence of the start codon or the AUG is methionine. Okay, now what about the amino acids that contain acidic groups? Okay, so we have aspartic acid or aspartate, we have asparagine, we have glutamate or glutamic acid, then we have glutamine. Okay, so these are the amino acids that contain an acidic group. Okay, now what about the basic amino acids? Now I hope I don't give up my I don't give out my age, but are you familiar with this movie Shallow Hal? So H, A, L, histidine, arginine, and lysine. So instead of shallow Hal, make it basic Hal. So the basic amino acids is Hal. Okay, histidine, arginine, and lysine. Now question, which of these amino acids is the precursor of nitric oxide? Which of these amino acids, which of these basic amino acids is the precursor of nitric oxide? Very good. It is arginine. Arginine is the precursor of the very potent vasodilator nitric oxide. Okay, now histidine is the one that undergoes decarboxylation and it becomes the very important vasoactive amine, which is histamine. Plays a very important role in acute inflammation, and it also plays a very important role in hypersensitivity reactions. Okay, and arginine also plays a very important role in the urea cycle. Okay, now here's lysine, arginine, and histidine. Now, what about the amino acids with an aromatic ring? So we've mentioned phenylalanine, we mentioned tryptophan, we mentioned tyrosine. Now, tip, histidine also has an aromatic ring. Okay, histidine also has an aromatic ring. Now, which of these amino acids is the precursor of thyroid hormone? Which of these amino acids is the precursor of thyroid hormone? Okay, very good. It's tyrosine. Now, which of these amino acids is the precursor of catecholamines, such as epinephrine, norepinephrine, and dopamine? Okay, it is tyrosine. Okay, so catecholamines. Now, which of these amino acids is the precursor of vitamin B3 or niacin? Which is the precursor of vitamin B3 or niacin? Okay, very good. Tryptophan is the precursor of niacin or vitamin B3. Okay, very good. Now, what is the amino acid that contains an imino group? Okay, it is proline. Okay, proline. Now, proline is a very, very important amino acid which causes collagen kinks. Okay, it causes collagen kinks. So please take note of that. Okay, that's proline. Now here, two atypical or non-conventional amino acids, pyrrolysine and selenocysteine. Question, which is known as the 21st amino acid based on its pK values? What is the 21st amino acid? Very good. It is selenocysteine. Okay, just remember the famous singer Selena barely reached her 21st birthday, sadly. Okay, now, almost done. So here's your guru guide, your stop codons. What are your 3 stop codons? Okay, so stop codons are very important, okay, in cutting short the length of the protein chain. So we have the 3, UAG, UAA, UGA. So UWAG, UA, UGA. UWAG, UA, UGA. So the stop codons. Now, what type of mutation do you have when a stop codon is involved? Remember, a stop codon will cause a premature termination of the genetic code. So what type of mutation is this? This is known as a nonsense mutation whenever a stop codon is involved. So there's premature termination of the genetic code. So just remember this, stop talking nonsense. Okay, stop talking nonsense. Okay, so I hope everyone got that. Now, winding up, I want everyone to bring this with them to your respective exams. What are the essential amino acids? So this is our famous mnemonics, Private Tim Hall or Private Matt Hill. Okay, so these are the essential amino acids. Okay, make sure you memorize this. The following are essential amino acids, or the following amino acids must be provided in the diet, except. So remember, essential amino acids, the human body cannot synthesize them. Therefore, these amino acids must be provided through the diet. Okay, this will come out in standard exams. Now, tip, always remember this mnemonics. Private Tim Hall always argues, he never tires. Therefore, in Private Tim Hall, the letter A here is what? What amino acid is the letter A in the mnemonics Private Tim Hall? What amino acid? This is arginine. So what does that mean? Private Tim Hall always argues it is always arginine. And it never tires. So what is the letter T here? What are the 2 letter T's? What are the 2 letter T's in the essential amino acids? We have threonine and we have tryptophan. It is never tyrosine. So always bring this with you to the exam. Private Tim Hall always argues, never tires. Okay, always remember that. Got it? Okay, so I hope you guys got that. So take a deep breath. Okay, we're going to end with this concept. What are the purely ketogenic amino acids? There are 2. It's leucine and it is lysine. So I want you to remember the word kill. K-I-L-L. Okay, so the purely ketogenic amino acids is leucine and lysine. So this will end our coaching session about the must-knows of the amino acids, and congratulations, you just finished one of the most essential chapters from the mother textbook in biochemistry, Harper's Illustrated Biochemistry, the chapter on the amino acids.